bispecific antibody format design is a strategic decision that balances multiple scientific and development considerations throughout the therapeutic lifecycle. Successful bispecific antibody design requires balancing multiple factors such as biology. The mechanism of action should guide the molecular architecture. Pharmacokinetics. The format needs to support the desired exposure, half-life, and tissue distribution. Production feasibility. The bispecific should be straightforward to express, assemble, and evaluate during early discovery. Let's begin with biology. Every format decision starts with one question. What must this molecule do to produce its therapeutic effect? This defines the structural requirements of the bispecific antibody. For example, in a T-cell engager, the bispecific antibody must bind CD3 on the T-cell and a tumor antigen simultaneously, bringing the two cells into proximity. Epitope distance influences format selection, as the architecture must support proper immune synapse formation. In dual receptor blockade, valency and epitope orientation must prevent unintended receptor activation or clustering. Scientists also consider whether effector functions such as ADCC are required, since FC design determines Fc gamma receptor engagement and cytotoxic strength. We also need to consider pharmacokinetics, as it determines circulation time, exposure, clearance, and distribution patterns. For example, fragment-based formats may improve tissue penetration, but often clear rapidly and require frequent dosing. Fc-containing, IgG-like designs extend half-life through FcRn recycling, supporting durable exposure, though their size can affect distribution. To improve fragment-based formats' half-life, engineering strategies such as Fc integration, albumin binding domains, and engineering such as adding disulfide bond to scFv can help balance exposure with tissue accessibility. Production feasibility matters in bispecific discovery. Some formats may look strong biologically, but face challenges with purity, pairing, and scalable production when moving towards CMC. Assembly efficiency is one key factor. IgG-like 4-chain bispecifics can face light chain mispairing, and the resulting byproducts are difficult to remove. Optimized chain ratio and purification strategies may help reduce these byproducts, while format design can promote correct chain pairing at the source. Pairing-controlled architectures, such as the Fab replacement strategy in WuXi Body, enforce correct assembly by replacing the CH1Cl with engineered domains derived from TCR beta and alpha, therefore eliminating light chain mispairing. Platform compatibility should also be considered early. Both CHO and HEC-293 are used for bispecific production, but they differ when transitioning to scale-up. CHO remains the gold standard for manufacturing, offering lower cost and reduced risk. For formats that rely on HEC-293, teams should consider early redesign for CHO compatibility or accept potential scale-up challenges. The optimal bispecific format is not necessarily the most innovative. It is the one that aligns biology, pharmacokinetics, and production feasibility from the start. To explore practical considerations for bispecific format design, visit the blog below and follow us on LinkedIn for more insights.